Yeah. At the outset, I thank the organizers for giving me this opportunity to present my topic on the efficacy of antibacterial compounds isolated from seaweeds for controlling Aeromonas Salmonicida infection in Oreochromis Mossambicus. Now, aquaculture is a key component of animal health industry with about 80 million tons of the annual fish production. The production increases year by year due to its importance in the human diet. The fish, what I'm presenting here, the Oreochromis Mossambicus, is an edible fish that is consumed by most of the humans in India and other parts of the world. The major concern here is the bacterial infection that is causing in the fishes, and especially the subsequent harm that is affecting the farmers, the production, in case of production. There are other bacteria like Aeromonas, Pseudomonas, Edwardsella, which are the major bacterial infections in fishes. The aquatic environment that is favoring for the pathogen to grow is this. One, because of the water quality parameters, improper nourishment, physiochemical parameters, the increase or the decrease in the pH, temperature, and overcrowding, and sudden change in temperature, and physiological changes. And as a result of all these things, the fish is subjected to reduced intake of feed, weaker immunity, and thereby causing infections and disease mortality. And the alternative that I'm talking about here is about seaweeds, which is rich in bioactive compound. Usually antibiotics are being given for treatment of bacterial infections in the aquaculture. But however, the use of antibiotics has been banned, or the indiscriminate use of these antibiotics can cause resistance. So to find out an alternative, we have studied the seaweeds as a source of antibacterial compound to treat these infections. They are low-cost alternative antibacterial derived from natural sources. These are found in the marine environment, and they are used in aquaculture for the most significant because of the versatile characters. They are effective alternative to be used of synthetic drugs instead of antibiotics, and their use in aquaculture. The objective of my study will be the screening of the seaweeds for the antioxidant and antibacterial activity, identification and characterization of bioactive compounds from seaweeds, pathogenicity of Aeromonas Salmonicida in Oreochromis Mossambicus, formulation of bioactive compound against the disease-causing pathogens by in vitro and in vivo method, and the most important thing that I will be presenting here is the encapsulation of bioactive compound as a fish feed. We have developed a fish feed which is incorporated with these bioactive compounds, and we have developed a patent for this, and the lab trial and the field trial. The methodology adopted here is, first we have collected the seaweeds, air dried it under shade, and it was extracted. The bioactive compounds was extracted using different solvents, the sequential and maceration method. Sequential is a hot extraction, and maceration is the cold extraction. And then we have studied the in vitro antioxidant and the physiochemical analysis of these extracts, then in vitro antibacterial activity, the separation of active compounds by chromatography, characterization and the identification of the active compounds, the toxicity of these active compounds, the formulation of the bioactive compound, pathogenicity and treatment by using these extracts, the changes in the biochemical and the immunological parameters, histopathological, and the encapsulation of bioactive compounds as a feed. Now, coming to the first one, we have, as I told, we have collected and processed the seaweeds, extracted using different solvents. We have determined the physiochemical constituent, determined the in vitro activity. So the solvent that were used for both, that is sequential and maceration method, is petroleum ether, dichloromethane, chloroform, ethanol, and aqueous extract. Now, if you see, we have chosen two different seaweeds, that is Sargassum longifolium and Gracilaria foliifera. Now, these are the phytochemical analysis of these compounds, Sargassum longifolium and Gracilaria foliifera. Among the 15 tested, carbohydrates, tannins, flavonoids, glycosides, triterpenoids, phenols, fumarins, proteins, and amino acids were present in both the extract, which has got good physiochemical characteristic or phytochemical characteristic of the seaweed. Now, there are enzymatic and non-enzymatic assays that we have studied: the superoxide dismutase, catalase, glutathione peroxidase, and other assays, and the antioxidant assay: total phenolic content, flavonoid content, antioxidant, reducing power, DPPH, ABTS, metal chelating, hydroxyl radical, and hydrogen peroxide assay. You might be asking, why am I focusing on all this? Because the DPPH, ABTS, antioxidant are all in relation with the ROS mechanism. And if the disease is or the infection is occurring in fishes, how does this ROS mechanism act? How does this defense mechanism is acting? That is where the basis we have studied to find out if this bioactive compound is active or not. Now, for that, we found out, we have taken all the extracts. You can see from my screen, we have selected all the extract, that is petroleum ether, dichloromethane, chloroform, ethanol, aqueous, of Gracilaria foliifera and Sargassum longifolium by the two different methods, and tested for the phenolic content. And we have found out that the ethanol extract has got good activity or good polyphenol content, when compared to the other extracts, total polyphenol, total phenolic compounds, and total antioxidant is also the same. The sequential method, that is the hot extraction, we found out that the total antioxidant was higher in the ethanolic extract of Gracilaria foliifera when compared to the ethanolic extract of Sargassum longifolium. Though the others had activity, the ethanolic extract of both the seaweeds have got higher activity. We have studied the total antioxidant, that is the hydrogen peroxide activity, then the DPPH, the radical scavenging, hydrogen peroxide, all were found to have good good ROS mechanism when compared with the others, that is the Sargassum longifolium. The Gracilaria foliifera was found to have high activity. Now, we've done an initial study to find out the in vitro activity by using Gracilaria foliifera and Sargassum longifolium. This is the plate method where we studied to find out if the compound is active against Aeromonas hydrophila. We have found out, yes, the compound, the Gracilaria foliifera, that is the ethanolic extract of Gracilaria foliifera, had some activity, which is clearly seen here by the zone of inhibition by well diffusion and disk diffusion. The well diffusion method of the ethanolic extract of Gracilaria foliifera showed 13 millimeter zone of inhibition, and Sargassum longifolium showed 8 millimeter zone of inhibition. And later we separated the active compounds from these seaweeds. Active compound from the ethanolic extract of the two seaweeds was separated by column chromatography. The crude extract were added to the column after the pre-elution of the mobile phase solvents. The active seaweed fractions from the ethanolic extract was achieved, and fractions were collected. And then we tested if these fractions are really having the activity. Yes, it had the activity against the Aeromonas hydrophila. The purified fractions of the ethanolic extract of the two different seaweeds were collected and tested for against Aeromonas hydrophila. Comparative zone of inhibition was found to be significantly high, between 12 to 13 millimeter in all the sequential fractions. That is, as you can see, this is a plate 1 of Gracilaria foliifera sequential extract. Plate 2 is Gracilaria foliifera maceration, and Sargassum longifolium by maceration and sequential. So if you find out of all these plates, you can see that the active compound, that is a purified compound, had a zone of inhibition, had an activity against Aeromonas hydrophila, which clearly showed that the compound is present, that the compound is acting. And preliminary phytochemical analysis of the seaweed extract were confirmed by the presence of the phytochemical, antioxidant, antibacterial activity of Gracilaria and Sargassum revealed that the ethanolic extract was higher. And then we wanted to find out what is that compound that is really acting upon this. So we did the minimum inhibitory, we found out the active compound, we characterized the active compound. So what we did here was, we did a combinatorial effect of the two seaweed. We combined the seaweeds of Gracilaria foliifera and Sargassum longifolium to find out whether it has really got a good activity in the ratio 1:1. The synergistic formulation of the seaweed was tested against the bioactive compound, and the results of the synergy was found to be effective in controlling Aeromonas hydrophila. This is the combined seaweed. This is the standard bioactive compound which we purchased and then tested. So if you could see, there is similar activity here, 18 and 32 millimeter. And the MIC, the minimum inhibitory concentration of the purified extract, was also tested, and tetracycline was found to be effective against the antibiotic in controlling Aeromonas hydrophila at 4 ppm. In the case of ampicillin, it was 10 ppm. But however, we found out that the extract was active at even the lesser concentration, right? At even 4 ppm, it was active. It was 4 ppm was much enough for the compound to act against Aeromonas hydrophila infection. In other words, to control the Aeromonas hydrophila infection. The GCMS was used, NMR, FT Raman, and HPLC was used to identify the compounds. So this is my GCMS profile of the ethanolic extract of Drasilaria florifera, Sargassum longifolium, and these are the compounds that are found in Drasilaria florifera and Sargassum longifolium. If you could see, there are 8 different compounds that we have identified with GCMS. And the synergistic compound, that is what I told about the mixing of these 2 compounds in the ratio 1:1. And these were the compounds that were found, phytol, and the 8 compounds were identified, phytol, tetradeconic acid, then octadeconic acid, squalene, butylene, etc. And we have also studied about the NMR activities. Then we've studied about the HPLC. These are the NMR activities, that is the peak shift between 1 to 1.06 have the peak difference, and peak to between 3.45 to 3.55. The NMR analysis of the combined seaweed extract showed a number of peak shift, which means it had a number of active compounds. The results reveal the presence of possible bioactive compound, which exhibited in GCMS study as compared with the standard chart. And FT Raman was also analyzed here. These are the compounds that we have identified, that is palmitic acid, squalene, butylene, then bromotaconic acid, then propylidine, then lipol. These were some of the compounds that were identified for FT Raman, and these were the active compounds. And one of these compounds, we have patented it and developed it as a feed. And these are the HPLC analysis of the compound that we've studied, or the bioactive compound. If you see, there are the retention time, there are a number of compounds that has been analyzed by the HPLC. And this is the HPLC chromatogram. This is my HPLC chromatogram. And the next, we have, having identified the bioactive compound, we need to find out whether this is active by in vivo method. We proved by in vitro, but are they acting by the in vivo method? For this, we have collected the experimental fishes, that is Oreochromis Mossambicus. Then we injected the pathogenic organism to find out the pathogenicity by 3 different routes, that is intramuscular, immersion, and oral route. And we have studied the treatment method. And initially, the physiochemical parameters were analyzed, and it was found to be at normal. And the intramuscular and immersion studies were found out that is 20 microliter of the pathogen was injected intramuscularly into fishes and absorbed for 120 hours for mortality. The concentration of 4.5 to 10 past 7 of cells, a viable cell, caused 100% mortality at 120 hours. This is without the treatment. The highest concentration caused 80% mortality after 120 hours. And it did not affect the fingerling when the bacterium was given through oral route. We observed mortality only by intramuscular and immersion. This is the LC50 and the LD50 value. So if you see, this is the control fishes. This is the symptoms or as a result of the bacterial infection. Damaged tail, pale gills. This is the pale gill. If you see at the control, there's nothing. You see here, pale gills. Then erythema, redness on the body surface. Then detachment of the scales. You do not find the scales here. Then redness on the tails. If you see between the control and the infected, there's a difference here. Then hemorrhage at the base of the fins and exophthalmia at the eye. And we've found out the tissue distribution by PCR method and found out that if you see at the lane 1 to lane 5, there's lane 1 and lane 2. That is the control DNA is lane 1. That is the Aeromonas hydrophila control DNA. And the gills had much of the bacterial content, much of the bacterial infection by PCR, and showed though the others were only very mild, the load was higher in the gills when we did a PCR. Now, these are the histopathological analysis. This is the control gills. The primary and secondary lamella clearly visible here in the control. However, in the infected by Aeromonas hydrophila, the primary and secondary lamella are damaged. And with respect to the liver, the glycogen vacuoles in the Cooper cells are clearly visible. But however, here it is damaged. Similarly, we can see the difference here for the skin and also by the control and the infected. And the physiochemical parameters were performed to ensure the water quality. Then Aeromonas Salmonicida were absorbed to be highly motile, cause high mortality in tilapia fishes. The PCR study confirmed the presence of pathogen distribution in the various organs. The last objective here, what I'm showing you, is the combined effect of bioactive compound against the disease-causing pathogen. Here we have developed the feed, the bioactive compound, and initially to test whether my compound has got the toxicity or not, we have tested first toxicity, and it was found to be non-toxic. The concentration of 100 to 1000 milligram per liter of the seaweed extract was found to be non-toxic. And then we did the treatment, the in vivo treatment. The in vivo treatment using the combined bioactive compound was performed against Aeromonas Salmonicida in Oreochromis Mossambicus. Aeromonas Salmonicida challenge fish, when treated with bioactive compound, showed 80% survival up to 15 days. And purified compound revealed potent disease-resistant capacity against the Aeromonas infection. If you see at my graph, you can see here, this is the control, which is having a lesser relative percent survival. And when the pathogen, this is the pathogen, and this is the treated. If you treat the treated, the relative percent survival is higher when compared to the others. And this is the feed that I was talking about, that we have patented it. This is the feed that is encapsulated with the bioactive compound, and this feed was directly given to the fishes rather than injecting into individually. And we have tested at the lab, and we have tested at the farm scale. It was found to be highly active. And this is the SEM analysis. This is the outer surface of my bead that I've taken, and this is the inner to find out, yes, whether the active compound is there or not. Yes, this is the inner surface. The cross-section analysis of the bead. The inside, no visible cracks or pores were absorbed on the outer surface. There's a clear round-shaped morphology of the feed. And the cross-section analysis showed the intracellular organelles, fiber matrix present in the bead. These are the active bioactive compound that is embedded in the feed, that is embedded in these beads. These are the active compound that is acting against these fish. And lab scale treatment, we did 3 groups. That is negative control, that is fish challenged with the bacteria and fed with the commercial feed. The group A, fish challenged with the pathogen and fed with the sodium caseinate bead, that is only the bead without the bioactive compound. And group B is fish challenged with the pathogen, fed with the seaweed encapsulated by sodium caseinate bead, that is a bioactive compound. And the results of the lab scale feed treatment revealed that the bioactive encapsulated feed is found to be effective in preventing the bacterial infection in fishes. In the treatment studies, 80 to 90% survival is observed, and it's eco-friendly and non-toxic. As I showed earlier, it is non-toxic. Compound is highly non-toxic up to 1000 milligrams per liter. And as you see from my graph, this is the positive control, the negative control, and the group A and the group B, which had highest percentage of mortality. This is the group B, which was treated with the seaweed encapsulated feed, or the bioactive encapsulated feed. And if you see at the morphology, that is the control, infected, and the treated. As you would have seen my previous slide, this is the infected and this is the treated. Absolutely, there was no loss in the primary and secondary lamella. There was no damage in the skin. There was no damage in the liver area. Cooper cells and glycogen vaccines were clearly visible here even after the treatment, right? But in infected, you can see there's a loss of these. And biochemical parameters, the amount of glucose triglyceride level was found to be comparatively lower than the carbohydrate in all the 3 groups. The estimated protein was found to be higher in control. Estimated level of protein was lower than the control in infected, and the protein levels is comparatively increased in the treated. Similarly, in the enzymatic, that is SOD, GST, and catalase, we found out that it was more or less similar in the control when compared to the control and the infected. The treated parameters were almost similar to the control. when compared to the infected. Large-scale treatment, we've done this in the field to find out if it is really effective in the field, positive and negative control, where this is a positive and the negative control. This is the experimental fish where we have done it in the open tank. If you could see the background, there's an open tank here. We've done it in the open tank and found out that the feed was given. The pellet was then put in the tank for the fishes to feed, and it was highly effective. To conclude my talk, bioactivities were identified from the seaweeds, were formulated as synergetic compound, tested for the toxicological response, challenged against the bacteria for the bioactive efficacy. Biochemical and enzymatic antioxidant parameters were improved after the treatment with the encapsulated beads. Bioactive compound encapsulated sodium caseinate was found to be effective in enhancing the growth parameters in fishes. These are the references, and thank you for your patient listening.